Welcome to The Explainer. Today, we're going to talk about a really powerful analytical technique, one that quietly makes sure that a ton of the products you use every single day, from your medicine to your food, are safe and are what they claim to be. I mean, think about it. It's a critical question, right? Well, the answer lies in this amazing method that can take a really complex mixture, pull apart all its individual components, and then tell you exactly what they are and how much of each is in there with incredible precision. So here's how we're going to break it down. We're going to follow what's basically an upgrade story. We'll start with the original concept, see why it really needed a serious performance boost, then we'll dive into the modernized tech step by step, and finally, we'll see how it actually impacts all of us in the real world. Okay, let's get right into it. It all kicks off with a fundamental scientific principle called chromatography. You've probably seen this without even realizing it. Imagine putting a drop of black ink on a paper towel, and then a little water touches it. You see the colors separate out, right? Blues, reds, yellows. That's chromatography. In the lab, it's the same idea. We have a material that stays put, the stationary phase, and a liquid that moves, the mobile phase. The different chemicals in our sample have a different stickiness or affinity for that stationary material. The stickier ones move slow, the less sticky ones travel faster with the liquid, and boom, they separate. Now, the original version of this, thin layer chromatography, or just TLC, it's a super simple, very visual method that's been a lab staple for decades. It's great for a quick and dirty analysis, but because it's so manual, trying to get precise, repeatable numbers out of it, that's really, really tough. But, science marches on. Industry and researchers started demanding more, greater accuracy, much faster results, and the ability to measure exact amounts. So this classic technique, it needed a major evolution. And that's where high-performance thin layer chromatography, or HPLC, comes in. Okay, so this is where it gets really interesting. Just look at the comparison here. HPLC is an upgrade across the board. It uses special plates coated with much smaller, more uniform particles, and the layer is thinner too. What does that mean? It means you get way more efficient separation. Instead of big, blurry spots, you get these super sharp, tight bands. And that higher resolution is the absolute key that lets us do accurate quantitative measurements, which was always the weak spot of traditional TLC. Yeah, so let's be super clear here. We're not talking about a minor tune-up. This is a complete modernization, a total overhaul. The high performance comes from automation that removes human error, from those finer materials that give us better separation, and from sophisticated instruments that let us see and measure things with a whole new level of clarity. So let's pop the hood and take a look. What does this high-performance process actually look like when you're running it in a lab? This whole workflow is really the heart of the upgrade. It's a highly standardized procedure where every single step is tightly controlled. You start by meticulously prepping the plate to make sure it's perfect. Then comes a really crucial step, that automated sample application. We'll get to that in a sec. After the separation happens in a controlled chamber, we get to the real magic, detection and scanning. This is how HPLC turns what you can see with your eyes into hard, precise data, making sure the results you get today are exactly the same as the results you'll get next week. And all this incredible precision, it's only possible because of a whole suite of specialized automated instruments that handle each part of the process flawlessly. First up, the applicator. This thing is a huge deal. Instead of a scientist trying to make a perfect little dot by hand with a pipette, this instrument uses nitrogen gas to spray the sample onto the plate in a perfectly straight, super narrow band. This creates the ideal starting line for that molecular race we talked about, which is absolutely fundamental for getting clean separation. All right. Next, the plate moves into a development chamber. And listen, a simple glass beaker just won't cut it anymore. These are specialized chambers designed to make sure the atmosphere inside is totally saturated with the solvent paper. This controlled environment gets rid of variables like humidity or temperature changes, ensuring the separation process is uniform and, most importantly, repeatable every single time. And finally, the absolute game changer, the densitometer. You can think of this as a high-tech scanner, but for molecules. It passes a very precise beam of light over each of those separated bands and measures exactly how much light is absorbed or reflected. Then, software takes that data and turns it into a graph with peaks. The area under each peak tells you the exact concentration of that substance. It's what turns a visual result into hard quantitative data. So, okay, we've gone through a lot of technical details. Why does any of it matter to you? Because this technology is a silent guardian, ensuring the quality and safety of countless products that we all interact with every single day. And just look at how versatile this is. In pharmaceuticals, it's making sure every pill has the exact right dose. In food, it can spot tiny traces of pesticides or toxins. When it comes to herbal supplements, it creates a unique chemical fingerprint to prove you're getting the right plant and not some cheap or even dangerous substitute. It's used all over the place, from forensics labs to the cosmetics industry. So yeah, the big takeaway here is that this isn't some obscure niche technique anymore. It's become an absolute cornerstone of modern analytical science. It's a reliable, cost-effective, and incredibly powerful tool for quality control and research. So what's the bottom line? By taking a classic technique and supercharging it with automation and precision engineering, HPLC gives us a fast, reliable, and powerful lens to see the true chemical makeup of the world around us. And it kind of makes you wonder, right? With a tool this powerful, what new compounds or maybe even hidden contaminants are we going to uncover next?